So our speaker for today's webinar is Dr. Cheryl Smith, who is an extension professor and plant health specialist at the University of New Hampshire Extension. In this webinar, Cheryl will discuss cultural and chemical management options for the most common and troublesome diseases of spring greenhouse crops. If you have any question during this webinar, please type it on the question box on your control panel. And after the presentation, my colleague Jason Lanier, who is helping with the webinar today, will read your questions and Dr. Smith will answer them. Without further ado, here is Dr. Cheryl Smith.
Good afternoon, everyone, and thanks for joining us today. I've got a lot of images and a lot of slides to go through, but do write down questions as we go through, and we'll try and get to those at the end. All right. So I'm going to go ahead and bring this up. I'm going to shut off my camera so you can actually see all of the slides. And
Cheryl, I don't think you have the screen, your screen out yet.
Good.
Yeah.
Okay. There we go. I think you had not given me permission yet. So.
Okay, yeah.
All right, so I think and my image is off of there, correct?
Yeah, the image is off. If you want to show it again, that's fine before you go on.
Okay. All right, so let me just back up one to make sure. So one of the first things that I wanted to mention here, I'm just trying to get rid of this panel on the side here. Let me get out of there. Okay, so update on disease management options for spring greenhouse crops. One of the first things I want to mention is, if I can get the slides to go, there we go, is that there are diagnostic labs throughout the Northeast. Every state has a diagnostic lab. If you want to know where the diagnostic labs are in the Northeast, go to nepdn.org, and there'll be a listing there of plant diagnostic labs with links to those labs. So you can find out where those labs are through this. If you are from outside of the Northeast region on this webinar, you can go to npdn.org and click on the region that you're in and find your local diagnostic lab to help you diagnose plant problems. I had indicated on the description for this presentation, I'm one of the editors for the Northeast Greenhouse Management Guide, and a couple of others also involved with that. And so I've been doing most of the disease section with some assistance from Leanne Punt this year. Just wanted to go through just real quick a few things that are changes in the greenhouse guide. I'm not going to spend a lot of time on these. You'll see them as we go through some pesticides for the various diseases. These are new products that have been added to the guide this year. Artavia is an azoxystrobin product. EcoSwing is actually a plant extract that is a plant biostimulant. Grotto and Calmore are both copper products. Optigo is a trichoderma combination with two different species of trichoderma. That's a biological that we have as well. Pageant TR Intrinsic. We've had Pageant Intrinsic for a while, but this is a total released sort of a gas that can be released. We've got a new hydrogen peroxide, hydrogen dioxide product, Purpose Plus. Not new, really. Sulf Oil X is a mineral oil, but based on recommendations from Leanne as well as recommendations from growers, it's very good in terms of management of powdery mildew. And another biological product that we have is called Zeo. It's a Pseudomonas product. There's also been some name changes because of company buyouts, etc. So what you've used in the past possibly as Disarm is now going to be called Fame. Another one is Emblem is now Sperato. So Emblem is being discontinued. And then we also have what was known as Pre-Stop. It's a Gleocladium, so another biological organism, is now known as Lalstop G46. The company Lalamond Incorporated actually made the Pre-Stop, and then it was sold by somebody else, but they have taken that all back in-house now and are marketing their own products. Mycostop is yet to have the label change on it, but will probably sometime before the end of the year. Elude is now Fostrol. Cleary's has been owned for a while by New Farm Chemicals, and so they are actually taking all the Cleary chemical labels and changing them over to their own chemical labels. So all gets confusing after a while, but I just wanted to give you an update on those chemicals. The other thing is please don't forget that there's lots of good references out there, and specifically when we're talking about spring bedding plants, the reference on the left, the Compendium of Bedding Plant Diseases and Pests, is available from APS Press, apsnet.org, and so is the Ball Field Guide. Both of them, Marge Daughtry and Ann Chase, are authors. Actually, I'm not sure if Ann is an author in the Bedding Plant Pests and Diseases, but Marjorie certainly is. Okay, so let's get right into the meat of the discussion here. Some common diseases in spring plant bedding plants. All right, so Botrytis blight. I can't imagine that anybody listening to this call that is a grower has not ever seen Botrytis blight. Causes a number of different problems. Some of the most common are things like this that we're seeing in this image, which is it's a blight, might start out in the senescent petals and then drop onto the leaves below, and any of that senescent tissue is good sites for colonization by Botrytis, and it's perfectly happy to move right into the healthy tissue. Can also cause stem cankers and cutting rots. In this case, here's a stem canker on a basil plant that is Botrytis. Cutting rots, the cuttings can often rot from Botrytis. And then one thing that's common with this Botrytis blight, it's also known as gray mold because it develops these masses of grayish spores on the infected tissues, and those become more and more visible with the higher the relative humidity gets in the production area. It also can cause spotting or necrotic areas on blossoms, any of the flowers that it can infect. So it has an extremely wide host range. It primarily attacks young succulent tissue, wounded areas, or older plant parts, particularly plant parts that are close to senescing. It's favored by temperatures of 55 to 70 degrees Fahrenheit. It needs about 6 to 12 hours of leaf wetness, and it really likes to have relative humidity of 93% or greater. One of the things that I mentioned is that any of the senescent tissue that's around certainly can allow the Botrytis to infect that tissue, and then it'll move right from that tissue into healthy tissue. So here's an example of a petal that fell off of a geranium onto the leaf below, and that's a new infection site. So any type of organic debris, deadheading not only encourages new flower production, but it helps to reduce the amount of Botrytis that's present. Here's another slide that Margie provided in this case on New Guinean patients with a lot of debris and then new infections occurring, particularly you can see in this leaf in the upper right. This also kind of emphasizes if you have hanging baskets over other plants on a bench and those hanging baskets are shedding plant material, it increases the likelihood that that plant material below those hanging baskets may develop Botrytis infections. Okay, so the management. One of the key management for Botrytis is to improve air circulation to reduce relative humidity. Plant spacing early on in the season really helps. I realize that as we get into, around Mother's Day or into April, early May, plant spacing can be an issue, but certainly reduced air circulation and increased humidity favors the development of Botrytis and powdery mildew. You'll see in just a moment. One of the things that we recommend, and that is also in pretty good detail in the greenhouse guide, is to use heat and vent cycles. So you open the vents, turn the heat on, that drives out the very humid air from the greenhouse, particularly as we go into evening so that you're not getting any condensation formation. Horizontal airflow, again, there's great detail in the New England recommendation guide about how to set up that horizontal airflow, number of fans per square foot, etc. And try to avoid watering late in the day, which increases that relative humidity over the evening. If you can avoid overhead irrigation, that's also helpful because you're not getting the foliage wet, or water early in the morning, and hopefully that has time to dry before the evening. Sanitation, get rid of that infected plant material, put it in a bag. Don't walk through the greenhouse with a plant that's infected with botrytis, because you're releasing botrytis spores all over the place. And fungicides, and it's really critical to rotate classes of fungicides based on FRAC code. And again, the greenhouse guide does list the FRAC codes in the main tables in the beginning. Horizontal airflow, okay? Bag it. Some fungicides, I'm not going to spend a lot of time on these slides, but this gives you an idea that botrytis has been around for a long time, and there are a lot of products that are effective against that. When we talk about rotating products and their mode of action, it's these numbers that are in the brackets off to the right that indicate the FRAC code or fungicide resistance codes. And so you're rotating between products that have different numbers, okay? The top 4 possibility for developing resistance development, so really critical to rotate out of those. There's also a number of combination products, and notice that Broadform, Orchestra, Mural, and Paget Intrinsic all contain a group 11 fungicide, so try not to rotate between those products. Lots of biologicals that are registered for this that are pretty effective as well. So please think about incorporating some of these into your toolbox as a rotation partner for some of our more conventional pesticides. Here's an example. This one is Botrytis. So this is a biological and actually gives pretty much better control than Daconil, or as good a control as Daconil. Both of these are actually much better control than Daconil. So it does work. So biologicals do give you an option. Powdery mildews. Same symptoms that you see on all hosts. It just looks like a light bit of powder, white areas. We'll see in a couple slides that there are some additional symptoms, some reddening sometimes may show up, but the symptoms are similar on all hosts. One thing I will point out is that no free moisture. So unlike botrytis, powdery mildew does not need to have a wet leaf surface to cause infection. It only requires a high relative humidity. So like botrytis, it's favored by high relative humidity. Most of the powdery mildews, however, are fairly host specific. So the one that happens to be on zinnia is not the same one that is on roses, etc. There are a few that have cross-host susceptibility, and I'll mention a couple of those in a moment. The infected leaves sometimes may brown and die. The symptoms and signs can be subtle. There's an example of on sedum. Powdery mildew is very subtle and looks more like a scab problem. So let's take a look at it. Speaking of the powdery mildew on sedum, this is what it looks like. So it looks like scabby lesions, particularly on the undersurface of the leaf, and it takes looking with a hand lens, getting it in the right light to actually see some of this mycelium on the bottom from the powdery mildew. It really doesn't form those nice white powdery colonies on sedum like it does on some other hosts. Another one that's kind of tricky to diagnose, and this is really showing a lot of powdery mildew spores and mycelium on the surface of these rosemary leaves. But when it first starts, you may actually see the leaves starting to brown and get necrotic and die, possibly drop off the plant. So it's very common on rosemary, but may be very subtle. Here's another one. In this case, hydrangea with powdery mildew. And this one is showing that the powdery mildew causes a reddening of the leaves. We'll see powdery mildew colonies underneath, and again, a hand lens and getting it in the right light is going to be helpful to try and diagnose it. But when I see this kind of reddening on a hydrangea, one of the first things I want to do is look for powdery mildew. So here's an example of something that has cross-host susceptibility. Powdery mildew on squash is the same powdery mildew that occurs on verbena. So if you're growing squash transplants in the greenhouse with your spring bedding plants, you can have powdery mildew move between the two crops. And it's also the same powdery mildew that gets on petunia. Okay, the chlorosis that you're seeing in the leaf on the right is actually due to powdery mildew infections. You can kind of see those powdery mildew infections a little bit better than the leaf on the left. But again, all three of those hosts get the same powdery mildew. So trying to not grow those vegetable transplants along with these crops will help prevent powdery mildew, or at least grow a vegetable transplant, something like squash that is powdery mildew resistant. That's really advisable. So the management, similar to Botrytis, you want to reduce that relative humidity. You want to improve the air circulation, those heat and venting cycles, horizontal airflow, same thing, water early in the day so plants have a chance to dry out and reduce the relative humidity. Sanitation's pretty critical too. You want to get rid of all the plant material, and particularly at the quote unquote end of the season or between crops, because the powdery mildew spores can certainly stay on that crop plant debris. It's really critical to rotate classes of fungicides or modes of action for powdery mildew. This particular fungus is notorious for developing resistance to, and resistance fairly quickly, to fungicides. Jeffrey, I think we might want to ask a question at this point.
Okay, so this is the first poll question that we have, and so take time and answer that question. So if everybody could answer that question, that would be great, so that we can move on with the next. Okay, so I'll close the question. If you still want to answer the question, please do. I'm giving 1 second to answer, then I'll close the question. Okay, Cheryl, can go on.
All right. So I want to talk a little bit about some of the fungicides for powdery mildew. And again, those with an asterisk on the top 3 in particular are at risk for resistance development, particularly the strobilurine fungicides. You really need to rotate those, but they are very effective against powdery mildew. So things like Pageant Intrinsic, Heritage, Artavia, one of the new products, they're all very good against powdery mildew, but you really need to rotate them. And sometimes it's good to rotate them with something like a copper, or a chlorothalonil product. Again, because those are multi-site fungicides with very low resistance development risk. There's some combination products out there, and notice that every single one of these combination products is a strobilurine combination. So you really should not rotate these combination products either with a strobilurine or amongst themselves, okay? And one of the things, again, I'm going to emphasize that biofungicides, yes, they work. They might be a little bit less effective than a conventional fungicide, but they can reduce the disease effectively, particularly for the powdery mildew. Things like actinovate, cease, triathlon work on powdery mildew, and it's great because the fungus is right there on the surface of the leaf. So these biofungicides are very effective against powdery mildew. So here's some of those options. And these are all Organic Material Review Institute approved, or organic approved. And again, that Sufoil X and the E's next to these mean that they're excellent in terms of their management. Don't forget that sulfur has been around for years and years and years, and is a very effective fungicide for use on powdery mildew. And then we've got a number of other products that are actually living organisms, or the extract from plants, as in EcoSwing or Regalia, that turn on the plant defense mechanisms. Here's another example of some of these products. So if we're looking at this slide, and again, these are from March Daughtry. This was a study done in 2006. The purple is not treated. The red is actinovate and capsule of wetting agent, then Rhapsody and the wetting agent. The yellow is strike, and what you can't even see there is compass alone, basically no disease development. Actinovate with compass, alternated with compass, no disease development, and Rhapsody, one of those plant stimulants, with compass, minimal amounts. So again, these biological organisms do work in terms of management, particularly for powdery mildew. Here's another one, just looking at rosemary, powdery mildew. And down at the bottom in purple and pink, we've got millstop alternating with cease, and we've got a horticultural oil alternating with millstop and alternating with cease. And then right down the bottom, oil alternating with millstop. So again, works on powdery mildew. Those products are all approved for use on an edible crop as well. Talk a little bit about downy mildews. Downy mildews are something that we see quite frequently on bedding plants, and it's one of those organisms that sometimes can get mistaken for other things. Common hosts that we see downy mildews on, we see it on basil. Needless to say, that's been a big issue for a number of years, basil downy mildew. I'd have to admit that with this year being as dry as it is, the outdoor crops, we did not see as much basil downy mildew this year, but phlox, potentilla, veronica aster, coleus is one that, because of some of the color patterns on coleus, it's very hard to see that on coleus. Argyranthemum is one that we can also get some growth distortion with powdery mildew infection. And then pansy, osteospermum. So let's take a look at some of the other indications for downy mildew. It has a relatively narrow host range. So a downy mildew that attacks a particular host plant has a pretty narrow host range and may only attack that particular type of plant. One thing, downy mildews are usually limited by the larger veins in the leaf, so they tend to have an angular appearance when you look at those lesions rather than round. Most of the downy mildews tend to sporulate on the underside of the leaf, so you might see a necrotic area on the top of the leaf or a yellow, angular lesion. Look on the underside for the downy mildew growth. A lot of these leaves will get infected and they may brown up and die, and it can be seed-borne or carried on the plug. So in the case of basil downy mildew, it is definitely seed-borne. So here's pansy with just a slight yellowing that's showing here in the center of the leaf. And if we were to look at the underside of those leaves, there is this purplish, almost sort of a purplish granular appearance to it, and that is all sporulation from pansy downy mildew. Here's downy mildew on potentilla, showing a nice example of those angular leaf spots, those chlorotic spots on the leaf, those yellow areas. If we flip that leaf over, we can see the nice fungal growth on the underside of that potentilla leaf. Downy mildew on coleus, I mentioned, is kind of a hard one to diagnose. In the image on the left, we are looking at a little bit of kind of a browning or discoloration towards the tip of that leaf. And if we pick that leaf off and look at the underside of it, it's got a very light sporulation on the underside of that leaf. Here's downy mildew on perilla. So again, with the color pattern on this perilla, it's very hard to see, but what we see on the upper surface, again, is sort of a discoloration and necrosis of the leaf. And when we flip that over, this brownish granular growth is actually all of the sporangia from the downy mildew fungus. Here's one in case of osteospermum and then also argyranthemum, where the downy mildew not only causes discoloration of the leaves, but it also causes a growth distortion. Particularly on the argyranthemum in the lower left, the youngest growth is very distorted, almost looks a little bit like an herbicide injury. Management for downy mildew, we want to improve the air circulation. High humidity actually favors the development of the sporangia, which are the reproductive structures that spread around the greenhouse with air motion and air movement. If you can keep the relative humidity below 85%, that's unfavorable for that spore to develop. If you can water early in the day, especially if you have overcast and cool weather, which favors downy mildew, again, sporulation. As soon as you find infected plants, because downy mildew produces numerous spores and it's a fairly difficult fungus to control with fungicides, you've got to remove those infected plants. If you're seeing sporulation, it's really difficult to get control of downy mildew. So you really, really should remove and discard those infected plants. There's a number of fungicides that are effective for downy mildews. The one thing, because the sporulation is occurring on the underside of the leaf, it's really critical to get coverage on the underside of the leaf. So you've got to somehow get spray up underneath those, which is why if you're seeing plants with a lot of sporulation, it's better to get them out of the greenhouse and then treat the remaining plants. The phosphites, things like phosphorol, oxyphos, very effective against downy mildews. And then a number of other products are also very good. Phosphatal the Aliette, and Areca are good products as well. Adorn has very good efficacy as well. The strobilurins are also effective against it, and several others on this list. Combination products, there are several out there, but don't forget that we've still got some pretty effective products with mancozeb and coppers. They're good protectants and they work well. They're not a curative, but they are a good protectant. Our hydrogen dioxide and hydrogen peroxides, such as these listed, X3, oxyphos, Zerotol-2, are also effective against the downy mildews. So biological products, we have several that are effective against downy mildew as well. The Bacillus amyloquiescens products such as double nickel, Stargus, and triathlon are all very, very good against this, as is Regalia. The one thing I'm going to mention about Regalia is it is a plant activator, so it is not a curative. Matter of fact, none of your biological products are really curatives. They really have to be applied prior to seeing disease or at the very, very first hint of symptoms of disease. With Regalia, it takes a while for that plant defense mechanism to be turned on and requires repeated application. So that really needs to be started early on in the crop, okay? And Jeffrey, we've got question number 2.
Yeah, so question number 2. Take a moment and answer that question, please. Thank you for those who have answered the question. We'll close the question.
All right. Moving right along here. All right, so here's some examples of downy mildew versus botrytis. So a lot of times it's possible to mistake one for the other, but again, look for those angular leaf spots. Downy mildew is a little bit more of a granular looking spore production, in this case on phlox, on creeping phlox. And over on the right is that botrytis. So botrytis is very gray, a little bit more powdery looking spores compared to the downy mildew. Downy mildew versus powdery mildew. So downy mildew on the top, you've got subtle symptoms. Look under the leaves, okay? So it's a yellowing that we see on the salvia on the left there, upper left. Underneath, we would see sporulation because there's kind of a subtle sort of mottling to those leaves, but the downy mildew is under the leaf. So it kind of reminds me a little bit of some of what we saw on the pansy. Powdery mildew is usually on the upper surface, but on verbena, it can have sporulation on both the upper and lower surface. But again, it's much more of a white spore that we're going to see with powdery mildew than with downy mildew. Bacterial leaf spots also cause an angular leaf spot. The bacteria is limited by the larger veins in the leaf. And in this upper left photo, this is a bacteria on columbine. If we look at the underside of those leaves, however, there is no sporulation. So particularly if the humidity is high, if we look at the underside of that potentilla leaf, we'll see sporulation underneath those angular leaf spots. Okay, and there's that example again. So moving into some root rot fungi, Pythium and Phytophthora, Rhizoctonia, Thalabiopsis, they are all root rot pathogens. The Pythium and Phytophthora are what we commonly refer to as water molds. They are favored a little bit by wet conditions and actually have a swimming spore stage. I'm going to concentrate on Pythium out of those. It's more common, although Phytophthora can be more devastating, but Pythium is a much more common problem. Rhizoctonia tends to cause damping off, can also cause a web blight on cuttings. We will not cover that one today. And then Thalabiopsis, also known as black root rot, is another one that's very common in a wide range of bedding plants, and I will talk about that one today. So in general, conditions that favor root rots are excess soluble salts, fertilizer, having a wet or waterlogged media, having a pH that is either too high or too low, and that pH that's too high or too low can make nutrients either available in toxic levels or deficient, which slows plant growth. An unhealthy plant is more susceptible to the root rots. Stressed plants, heat stress, fertility stress, drought stress can all make them more susceptible to root rots. And then cooler temperatures favor the development of root rots because it slows the growth of the plants. And we also see cool temperatures, particularly favoring things like the black root rot, the Thalabiopsis. Here's an example of, sort of looking for root rots is if we look at this photo on the left, if you look down this entire bed of geraniums, you can almost see a yellow sort of stripe down the middle. Needless to say, that center portion there closer to us, pots actually had very, very stunted plants, yellowed plants, very, very wet soil. What the situation was here is that this was a gutter right above this area, and that gutter was leaking. What the grower did was remove the plants from this center area because they couldn't fix the gutter right at that time with the plants on the bench. They drenched the remaining plants on either side of this quote-unquote yellow stripe, and then they fixed the gutter when they got this crop out of the greenhouse. So that wet soil condition really favored the development of Pythium root rot. So where does these root rot organisms come from? They can come on transplants, they can come on rooted cuttings. If you aren't good about sanitizing benches and tools and flats, if you're reusing flats, they can sit in any little crevice on the benches or tools. If you leave potting media bags or containers open, these pathogens can blow into that potting media. If you're storing potting media on the ground or outside and those bags rip, come into that. Our root rot pathogens can come in on seed. If you're working with infected plant material and getting rid of it, and then you don't wash your hands before you're working with another crop or potting up a new crop, you can transmit that. Hose ends on the floor can pick up spores of these fungi. And then when you turn that hose on, it washes the spores into the pots. And needless to say, dirty floors with lots of plant debris, etc., can also be sources of root rot pathogens. So here's an example. On the left, kind of clean, not too bad, but there's still plant debris there, and that hose end on the floor, when that gets picked up and is used to water plants, it can carry spores. So better to keep the hoses up on the bench like this with quick connect sprayers that you can connect to those and keeping them clean. Some of the general root rot symptoms that we see are stunting, nutrient deficiency symptoms because the roots aren't functioning properly, so it can't take up nutrients. Wilting, again, can't take up water. And when you pop the plant out of the pot, if you suspect a general root rot symptom, pop the plants out and take a look to see how the roots look. Discolored roots are usually an indication that there's a root rot, and certainly waterlogged media because it's not using the moisture because the roots can't function properly. And in direct seeded crops, damping off is also an indication that you've got a root rot. So check for healthy roots. If you're seeing what look like nutritional deficiency symptoms, before you even start with a pour through, pop the plant out of the pot and see if the roots look healthy. In this case, the ones on the right the ones on the left look pretty good, but the ones on the right, very water-soaked, dark color, not healthy. This is Pythium root rot. Pythium root rot, one of the things that we see, again, all those general symptoms, stunting, nutritional deficiencies, the roots rot from the tips. So Pythium comes in at the tips first, works its way up, can work its way right into the crown, right into the stem, causing something called blackleg. One of the key identification factors is that the cortex sloughs off the root. If you were to tug on an infected root gently with your thumb and forefinger, you would pull that cortex off, leaving the inner steel of the root. So it's kind of like taking the paper sleeve off a straw. Many hosts, Phytophthora is more aggressive, but it's less common, and Pythium can also cause crown rots and stem cankers. So here's what I'm talking about when I talk about sloughing roots. So if we take some of these roots that are kind of grayish color and we gently tug on the root, on the bottom right, where that arrow is pointing, the cortex has been pulled off of that root and it leaves that inner steel. That's a diagnostic feature for Pythium and Phytophthora root rot. So if you found that, you're not dealing with Rhizoctonia, you're not dealing with Labiopsis. And in the picture, in the left-hand picture, if you look up towards the stem, this had advanced to a point where we also have what's called Pythium blackleg on this plant as well. So very advanced. Excessively wet media favors Pythium development, and high soluble salts, particularly ammonium nitrogen fertilizers, can burn the roots, stress the roots, and that favors Pythium infections. Steaming media with high organic matter. So soil media steaming used to be fairly common. It can release ammonium, which is toxic to the roots, and then can also result in Pythium infections. Fungus gnats, so if you have a lot of debris on the floor, the potting media is kept very wet, it can attract fungus gnats. They're attracted to the rotting roots. The fungus gnat larvae feed on those roots. They carry the spores within their guts, and they can spread that around. And even some of the adults can spread around spores as well. So key here is sanitation, really monitoring your electrical conductivity, managing both fungus gnats and shore flies. Shore flies can also carry them around in their body, and fungicide drenches. Lots of stuff is registered for Pythium. I'm not going to go through this list, but again, the phosphites are fairly good because they work pretty well on these water molds, and downy mildew is also considered to be a water mold. Combo products, a number of different ones. The hydrogen dye and peroxides as well are effective against these as a drench. There are some pretty good biological products for controlling Pythium root rot, and a number of these, the Lalstop, particularly the Bacillus amyloquiescens products like double nickel and triathlon are pretty effective. But again, these are much better as a preventative, not as curative. So your biologicals are really used as preventative, not as curatives. Just an example, so Uptego, again, is one of our, let me just back up, I'll try and go backwards if I can. So Uptego is a trichoderma, 2 species of trichoderma, and Uptego treated on the left on ranunculus, inoculated with Pythium, and untreated on the right. So it does give you efficacy. So the last root rot I'm going to talk about is the Labiopsis root rot, also known as black root rot. Again, all those general symptoms, stunting, nutrient deficiency. With the Labiopsis root rot, it's known as black root rot because the roots are stunted, and the Labiopsis has these really dark fungal structures that are in the roots, so the roots will often look blackened. It can often develop quickly on recently shipped plants because sometimes those plants are shipped at times of the year when they're exposed to cold. They tend to be a bit cold and they get stressed. They might dry out a little bit during shipping, and they tend to be more susceptible to Labiopsis root rot. So here's some symptoms of Labiopsis root rot. Pansy is one that's extremely susceptible to Labiopsis root rot, and it causes stunting, also yellowing. The image on the right, lots of losses due to Labiopsis in these plug trays. And here's what we're talking about when we talk about black root rot. So In the upper left, there's areas on the roots that are very dark and very black. If we take a closer look at those, we can see that they're patches, extremely black areas in the root. And in the bottom left, we can, with a, probably about maybe 40x magnification, we can see some of those black fungal structures. When we look at them in the lab microscopically, Deb, my lab assistant, and I kind of like to call them Tootsie Roll fungus because it sort of looks like Tootsie Rolls that you can break apart at little segments, and they can break apart in segments. And those spores, when they're dry, can blow around the greenhouse, the potting areas, and get into potting media pretty easily. Again, the arrows are pointing to blackened areas on the roots. A lot of times this gets mistaken, and you might end up thinking that it's Pythium, but again, you don't have the cortex that will slough off of these. It's a fairly difficult fungus to control and really requires good sanitation as well as fungicides. There's the photo of the Tootsie Roll fungus. So each one of those little segments can break off and blow around the greenhouse and cause infection. If the crop is stressed, if you have cool, wet media, if there's soil in the growing media, soil is extremely favorable environment for Thalabiopsis root rot. Not too many people mix soil in the growing media. We do have some growers that still do, but certainly that will increase the likelihood of Thalabiopsis developing. Excess ammonia and nitrogen, nutrient imbalance equals pansy problems and makes pansies more susceptible to black root rot. pH above 5.5 favors disease. So again, some of those crops prefer to have a little bit more acidic pH. So watch where your pH is for Thalabiopsis root rot. Anything that you can do to reduce plant stress will help manage Thalabiopsis. Make sure that your media is well drained, that you've picked a potting media that drains well for seedlings. The longer it stays moist, Thalabiopsis, and also Pythium. Make sure that you've got warm soil temperatures for plug trays. If it's early in the season, try some underbench heating or heat mats to keep that, avoid having cold soil temperatures. If you've got stuff that's blowing media and soil and stuff around the greenhouse or around the head house or potting area, these spores of Thalabiopsis are long-lived and can spread. Do not reuse pots and trays that you have had crops in that have had black root rot. It is almost impossible to wash this out of those pots and trays, and it's just advisable not to reuse them. Sanitation, 10% bleach and scrub, or you can use Zero-Tol or Green Shield and disinfect those benches for about 10 minutes. And again, here's those crops. Plugs, the one on the right and the one on the left are healthy, but the one in the middle is infected with Thalabiopsis root rot. Okay, so this grower was reusing these plug trays from crop to crop, and every year got increased mortality. When they stopped using these plug trays and got completely new and clean plug trays, they decreased the amount of Thalabiopsis they had by about 85%. By and far, the best product for controlling Thalabiopsis root rot is thiophanate methyl products, hands down, in multiple research, time and time again. All right. Others are labeled as well, but by far, thiophanate methyl products are the best. We do have biological products, but again, used prophylactically. So if you have susceptible crops like pansy, petunia, Calibrachoa is very susceptible. Think about using one of these biological products in rotation with a thiophanate methyl product. Or if you're trying to grow organically, these are your options. So again, just remember when you're trying to manage root rots, hoses off the floor, clean, clean, clean. Don't reuse those pots and flats. Get rid of the worst infected plants, and good management of fertility and pH. Okay, and disinfect cutting tools as well. All right, last thing I'm going to cover today is a little bit about virus diseases. And one of the viruses that we see quite frequently every year in bedding plants is tobacco mosaic virus. So it causes a mottling or a mosaic on the foliage, in this case on petunia. A couple years ago, probably more than a couple years ago now, it's probably about 3 or 4 years ago, we did see quite a bit of TMV on petunia. There was an outbreak of that from a couple suppliers. This is one of my favorites. Remember that I'm a plant pathologist and I get excited about diseases, and this one brings a smile to my face because I think it's absolutely beautiful. This is on monkshood, on aconitum, cucumber mosaic virus, which has a very wide host range, as does tobacco mosaic virus. But on monkshood, it causes this sort of beautiful yellow ring spot and kind of zigzag pattern on the leaves, and a flower break, a break in the color of the flowers. So you'll actually get ring spots and mottling on the flowers as well. And that shot there is just a scanning electron micrograph showing the virus particles in this case are more spherical. Western flower thrips. So thrips are you know, a bane of the greenhouse industry. Western flower thrips in particular are of concern because they are a vector for tospoviruses. Our two important tospoviruses are impatiens necrotic spot virus and tomato spotted wilt virus, which thrips both as adults, seen on the right, as well as the immature, seen on the left, can transmit both of these tospoviruses. Here's an example of tomato spotted wilt virus on tomato. So this particular photo is from last year. These were transplants that were grown in a greenhouse. Some bedding plants in the greenhouse, but a major thrips problem in the greenhouse. So these were brought outside to harden off, and they noticed spotting and then these sort of stem lesions developing on them. And there is a bit of almost a ring spot on some of the lesions on the leaves, and the modeling pattern made me suspicious of virus, and we did find a few thrips on them, so tested, and this tested positive for tomato spotted wilt virus. Also saw this at least 3 times this spring. In one case, the grower was growing high tunnel tomatoes. They had grown their own transplants and had started those transplants in a greenhouse with bedding plants. And there were thrips present. So those bedding plants had TSWV. The thrips transmitted them to the tomatoes that they were growing from seed. So these tospoviruses are not seed transmitted. So if you can keep seed grown crops separate from vegetatively grown crops, and you can keep thrips down, you can manage tospoviruses. This time, the line patterns and the zigzag here is a little bit different from the previous photos, a little bit more of circular patterns, maybe some ring spots. This one has black necrotic areas, not so much on the petioles, but this time is impatiens necrotic spot virus, but again, transmitted by thrips. Here's one that's a little bit harder to see. This is tospovirus, this is INSV, on verbena, and on verbena it's very subtle. It's got these light and dark green sort of mottled areas on the foliage. Thrips were present in this greenhouse, and there were some on the plants, so we tested it for INSV and it came up positive. Basil. Most folks are growing basil from seed when they're starting them, and if you're growing them in the greenhouse with bedding plants, there's a good chance if there are thrips present that tospovirus can move to basil, okay, and causes that kind of mottling and a little bit of ring spotting on the foliage. Here's another one, INSV on Nemesia. Again, thrips problem in this greenhouse and causing these ring spots and necrotic ring spots on the Nemesia. One thing that you can do in-house is there are immunostrips available from Agdia. You can buy them in packages of 5. You can run your own in-house immunostrip test. This is what we're using in the lab to test for these. I buy them in sets of 25. It's about a 10 to 15 minute test. So in this case, it's an impatiens with these black zone line patterns in that leaf that's in the upper center, and then black zone cankers that are on the stems, classic impatiens necrotic spot virus symptoms, or TSWV symptoms. And so you're going, which one is it? Well, if you run these tests, you can see that this is tomato spotted wilt virus, which causes a little bit more of these line patterns as opposed to a ring spot, which we would get from INSV. Be aware that sometimes diseases aren't by themselves. So in this case, we've got INSV and powdery mildew, both showing up on this begonia. The INSV is causing this weird sort of green mottling, and the powdery mildew is causing the reddening, as well as you can see some of the powdery mildew colonies on that leaf over at the left. Our pepper and other bedding plants that we grow for vegetable transplants also very susceptible to these tospoviruses. So this is INSV on pepper plants, a little bit of mottling and some chlorotic line patterns in the upper left, and on the lower right, necrotic ring spots. I most often see the necrotic ring spots when I see peppers that are infected with tospoviruses, usually INSV is what I find. And again, these are, you're growing them from seed, and you're growing them in houses that have thrips and bedding plants that are moving those tospoviruses around. A little bit different. This is another disease known as tobacco rattle virus. This is one that I cannot test for in-house, nor can you. We send these out to Agdia, and this one actually has to be tested with molecular means through PCR. So this is Hosta crassa regale with these white chlorotic patches on the leaves. And if you look at the leaf on the left, on the right, down towards the base of the leaf, there's a little bit of ring spot pattern there as well to the green. So that sort of suggested, well, is it possibly virus? I mean, this could even be mistaken for a little bit of spray injury, which is initially one of the things maybe one of the growers might have thought it was. But they also had a history with tobacco rattle, so we sent it out and got it tested. And any of the virus-infected plants that you have really need to be destroyed. You cannot cure a virus-infected plant. And certainly if you take cuttings from those plants, you are propagating a virus. So isolate the suspected plants and get them tested, or do the testing yourself if you want to go that route with some of those immunoscript test kits. Once you have confirmed infected, oops, sorry about that, misspelled that word, confirmed infected plants, those need to be destroyed. You cannot cure them, and if you keep them around, you run the risk of that moving to other crops and other plants. Some, like tobacco mosaic virus, can be moved around simply by touching the plants and then touching healthy plants. Aphids can move around things like cucumber mosaic virus, and then certainly thrips can move around the tospoviruses. So you really want to make sure that you manage thrips. And obviously no cuttings from infected plants, because that virus is in the plant even if some of those leaves are not showing symptoms. Jeffrey, question number 3.
Okay, question number 3. A few seconds before I close the question. Okay.
All right. So The last thing I just want to talk about is when you're sending in a sample to a diagnostic lab, whatever diagnostic lab that might be, is just realize that the quality of the sample that you send us is really going to have an impact on how we're able to do an accurate diagnosis. So make sure that you're storing that sample properly, refrigerate it until you can ship it or deliver it. Most of our labs are not taking in-person samples right now due to COVID-19. So you're best to ship those samples overnight. And for information, the UNH Plant Diagnostic Lab will likely be reopening sometimes within the next week or two. There will be a notice going out for that, so you'll be able to mail in samples to my diagnostic lab. Most of the others are already open and accepting mail-in samples as well. And I think that's all that I have. Here's my contact information. And you can get to the Plant Diagnostic Lab UNH by unhpdl.org. Also Facebook, UNH-PDL is the Facebook page. So I think that's all that I have, Jeffrey. And
Okay. So,
I'm going to go back and stop sharing. So
we have Jason look at the question. I don't think we have any questions.
Oh, okay.
Jason,
No, this is Jason. No, we don't have any current questions, but I'd ask anyone in the audience who has a question to please go ahead and key that in using the question function now, and we can pass that along to Dr. Smith.
There's one I can see.
Okay, Cheryl, question is, do fungicide applications kill biofungicides if they are alternated or in rotation?
That's going to depend upon the biofungicide. So the best thing to do is check the label of that product, or if you have a particular conventional fungicide that you're using and you want to use a particular bio with it, the websites of the companies that produce those bios are usually very good, but I'd also suggest contacting the company itself, because they're very good about knowing what conventional fungicides may react negatively with their products. Certainly a lot of the fungicides are very compatible with a lot of the bacillus products and also with a lot of the trichoderma products as well. But the best thing to do is check with the manufacturer.
Excellent. And the next question is for root rot diseases. Soil drenches are the best, correct?
In most cases, yes. Soil drenches are best, although you can incorporate a number of the bios come in granular formulations that can actually be mixed in with the media prior to either seeding or transplanting or up potting what you might be doing.
And as far as black root rot goes, the best rotation for black root rot in particular?
I would probably look at incorporating one of the biologicals into the potting media prior to getting the crop going, so that bio has some time to get established in that potting media. And then you've got that in there for particularly on susceptible crops. And then look at a rotation of perhaps a drench with a bio with thiophanate methyl.
Great. And so two more, and that's probably all that we have time for today. Rosemary leaves dying off. Could it be powdery mildew?
It certainly could. I've got a sample that I'm working on right now that has some slight distortion, some slight necrosis, and I was looking at them yesterday and microscopically and could not find a bit of powdery mildew on them. So that's still a stumper for me, but a lot of times necrosis can certainly be powdery mildew on rosemary. Kind of goes undetected.
Okay, and I think last one for this session, best practices during poinsettia propagation to avoid botrytis.
Spacing, reduce relative humidity, and make sure that you've got some good airflow. Fungicide, there are some fungicides that you can use, but again, you've got to be careful when they start to get up to bract color. But that spacing and airflow and relative humidity are going to be a couple of things that are really critical.